step 1 is the same as these people already mentioned about DNA extraction. DNA extraction is from, if you are doing it from a tissue layer or from blood, you could use certain methods that you use using SDS. And if you are doing it for bacterial DNA, what you do is you add EDTA combined with lysozyme, which digests the cell wall because the bacteria contains cell wall. So after extraction of the DNA, what you do is the next important process is the digestion using endonucleases. So restriction endonucleases, that means these endonucleases have the ability to go to a specific site in a DNA sequence and digest them from that point onward. For example, you have a sequence like TAG. So this endonuclease has the capability of going to that specific site and breaking the section fragment length polymorphism, that is for each individual the length of the fragments might differ. So it is due to these endonucleases that these occur. So after digesting them with endonucleases, now you have the DNA strands broken up into pieces. So the next process what you do is you carry out the gel electrophoresis in the presence of ethidium bromide. I did explain about the electrophoresis, you know what the procedure is now. So after doing the electrophoresis, we do a process known as depurination, denaturation, and then finally neutralization. Okay, so this I think you understand what you mean by denaturization and neutralization, but it is actually the breaking of the beta and glycoside bonds between purine bases and the ribose sugar. So you know the structure of DNA. I think you would be able to visualize the structure of DNA. So you have the DNA and the ribose sugar there, and the purine bases are joined to them. The breaking of the we call them the beta and glycoside bond is known as depurination. So what you do actually in this case is you add a solution containing low pH, for example an HCl solution. So once you do that, you could after treating with HCl, you could really break the bond between the nitrogen base and the ribose sugar. And depurination, this does not occur to all the nucleotides. It just occurs at certain locations. That is to make the fragments even shorter. For example, now we'll understand later when I'm explaining about the nitrocellulose membrane denaturation process. What do you mean by the denaturation is breaking the hydrogen bond between breaking of the hydrogen bonds between two nucleotide strands. So you can understand that you know that DNA consists of a double helix, 2 strands are there. So what do? You break the hydrogen bonds and separate the 2 strands into 2 regions. So that's what you do. You do it. That is what denaturation. So you know that DNA could be denatured in 2 forms, in an acidic phase and also an alkaline phase. An alkaline phase is more stable, so we do an alkaline denaturation here. After doing that alkaline denaturation, now you have to do a neutralization process, because these molecules of DNA will not bind onto the nitrocellulose layer if the pH is greater than 9. Now we did an alkaline denaturation here. So what we actually do is we are not going to add an acid and neutralize here. What we do is we add a trisalt buffer. So by adding a trisalt buffer, we do neutralize this. And that also would be explained later, because there is a difference between the as I said, the nitrocellulose membrane and the nylon membrane. So those differences should be explained later. So I'm just giving you a briefing of the steps right now. So as I said, depurination is accomplished by soaking the gel in acid. So and denature the DNA, and then you could understand what I meant. Okay, the third step. The third main step is the main concentration, the point to concentrate here about the transfer of these fragments on the nitrocellulose membrane or the nylon membrane. So usually they use the nylon membrane or the nitrocellulose membrane, because there are many disadvantages of the nitrocellulose membrane which will be explained later. So now what happens is, what we do is we do a process like this. What you do is, now you got a plastic support here. You got a plastic support here, and this is your agarose gel, the gel that you got it from the electrophoresis, and the denaturation and everything is done and complete. So what you do is you add a transfer buffer. This is known as a transfer buffer. So this transfer buffer just maintains the pH, the required pH for the DNA fragments to go and bind onto the nylon membrane. So this is the nylon membrane present over here. So what you do is this is a filter paper wick. what happens is this buffer moves through this. This buffer, what happens to this buffer is moves through this filter paper wick, and what happens due to capillary action, this moves up. So you have you can see here there are absorbent papers here and filter papers here. What this absorbent paper and the filter paper does is induces the capillary action. So what you do is there is a weight over there. So due to the weight, it applies a pressure so that more and more buffer transfers upwards. So not only the buffer is transferring, you should remember that the agarose gel is over here, and within the agarose gel you have the DNA fragments. So what happens is while the capillary action, while this buffer is transferred, little by little, what happens is the DNA from the agarose gel is transferred onto the nylon membrane. This is done for quite a period of time so that almost all DNA fragments you have in the gel is bound onto this. Understand how this procedure happens and goes and so fourth step, very important step, the hybridization step. So Anish explained what you mean by hybridization is. So now you have all the DNA fragments on the nitrocellulose layer of the nylon membrane. So we don't have anything there in the gel, everything is taken onto the nylon membrane or nitrocellulose membrane. So now, before that I'll just tell you what you mean by the hybridization is. What you do is, again you take this nitrocellulose membrane or the the nylon membrane, and you put it into another buffer, and then you add the hybridization probes. These hybridization probes are artificially synthesized nucleotide strands which have phosphorus 32, which is radio labeled. As we know this, as I said before, the DNA that we extracted onto the nitrocellulose membrane is also single stranded because we did a denaturation process, remember? So because of that, this is also single stranded, and the probe is also single stranded. What it does is it goes and hybridizes. So we know the sequencing of the nucleotide strand, I mean the probe that we are sending, we know the sequence. So that, for example, say you need to identify an insulin producing gene in the nitrocellulose layer. So what you do is you take an mRNA copy. You know how protein synthesis occurs, right? So you take an mRNA copy, and from that you derive a cDNA or a complementary DNA which contains the probe, which is radio labeled, and then you send it to the nitrocellulose layer. Then it specifically goes and binds on the location where you have that insulin gene. So you recognize the nitrocellulose, the exact position of that insulin gene in the nitrocellulose layer. So that's what this hybridization does. So any doubts on that? I was just clear with it, right? In this diagram you can observe the hybridization probes. You can observe that the probes are not attached to every strand. It's just attached to specific locations, so that you could really identify at which place your interested gene is. So that's the main thing that we are doing this southern blotting is to identify the place where the interested gene is. After the hybridization is complete, what you do is you just pass some, you wash it, and then you pass some X-ray onto it. So then the probes, the areas where the probes are hybridized would show up so that you can identify and extract the required gene of interest. So so the next thing I told you, the nylon membrane, the nitrocellulose layer. Nitrocellulose layers were used in the olden days, but now what we use is the nylon membrane. So the advantages of the nylon membrane is that the nylon membrane could be charged. For example, now you have a comb, you comb your hair, and you take a piece of paper and try the comb attracts the piece of paper. So the nylon membrane is something like that. It could be charged. So when it is positively charged, you know that DNA is negatively charged, right? So in that case, you could just make the process of binding of the DNA even more easier onto the nylon membrane. So it facilitates the binding of the DNA onto the nylon membrane even more easier. But if you are using a nitrocellulose layer, after it is extracted, you have to bake it at 80 degrees centigrade for proper attachment, covalent attachment onto the nitrocellulose layer. But nylon membrane, since it could be positively charged, you needn't do that. You can just just do the blotting, and the DNA comes and attaches.